Hello, everyone, and thank you for joining us. I'm Jack Andrews, the Vice President of Business Development for Kura Biotech, and I've been working in this industry for 10 years now. Kura is really happy to send a big thanks to the Alliance organizers for giving us the opportunity to sponsor this event. Now, you may have noticed that we have three 15-minute time slots, and instead of giving you three sales pitches from Kura, we decided to take a cue from the Alliance's mission and share some knowledge. We've put together a three-part series on the evolution of using beta-glucuronidase in urine drug testing. Think of this as a past, present, and future, or maybe today we talk about yesterday, tomorrow we talk about today, and Wednesday is in, and we will talk about the future at that point in time. So with that, I'll turn off the camera, see you guys at the end, and we'll talk about the evolution of beta-glucuronidase. So today's 15 minutes, we'll touch on who Kura is, very briefly discuss why we need to use beta-glucuronidase in our laboratories, look at some of the original enzymes, and then make sure to join us for parts 2 and 3 tomorrow and Wednesday, where we talk about today's current trends and what challenges we may face. Since the inception, Kura has always held the belief that drug testing is never routine, and our mission is to provide labs with the best possible molecules to make drug analysis simple, fast, and precise. So how do we accomplish this mission? We believe we have to follow our core commitments, which are unlimited spirit. For Kura, this means we define things by potential, not by limitations. Often this pushes us out of our comfort zones, but we believe that drives advancement. We have unapologetic standards, or we say there's no excuses. We are determined to challenge the status quo every day without impacting quality, because we know that if we fail, somebody else pays the cost. For us, human connections are all about not forgetting life is about people. Our employees, customers, suppliers, patients, partners, the whole community is impacted by our activity. So we strive to keep that knowledge present in our daily activities. This is a perfect segue to introduce our give back. Kura gives back 2% of our revenue. The employees of Kura actually make that give back. We don't look at this as charity, but as an acknowledgement that a strong community is vital to a happy life. So what specifically is Findon? Well, Kura Biotech is a biotech company with core expertise in making enzymes for analytical purposes. We have 3 areas of expertise: genetics, which is supported by a brand named Avenir; proteomics, supported by Blicka; and toxicology. Findon is the group of people dedicated to the tox world. Currently, we're present in more than 15 countries, and we're happy to provide enzymes to the world's biggest laboratories. And all that translates into about 2 million samples per month being tested in validated methods. Our ISO 9001 facility is the largest beta-glucuronidase producer in the world. Okay, let's get into some science. First, let's go over some very, very elementary metabolism. So Jack took some morphine, and then he has to send a urine sample to the lab. The question is, what's in the cup? Well, the liver gets involved and hooks a glucuronide on. Now the kidney is much happier to excrete that drug into the urine for elimination. In the case of morphine, the parent molecule is ingested, but 3 compounds make it into the cup: parent morphine, morphine 3-glucuronide, and morphine 6-glucuronide. Enter beta-glucuronidase. So when you combine this enzyme with the sample, you can clip off those glucuronides and convert everything back to the parent form. Well, you might say, our instrumentation is sensitive and selective. Why not look for all 3 compounds? Well, by definition, Looking for the parent and metabolites certainly increases the complexity of the method. And if you start to look at panels of molecules, that complexity can explode into numbers. Second, you'll need to make glucuronide controls for those molecules. Those can be hard to find, if not impossible in some cases. Also, glucuronide internal standards are even more difficult to find. So in pretty much every case we found, a glucuronidated material is much more expensive than the parent molecule. So this increases the cost to your laboratory. In short, this tried and true technique has proven to improve sensitivity and analytical robustness for many years now. So discussing the evolution of available enzymes. In the beginning, the products were very crude extracts from a variety of animal tissues known to express beta-glucuronidase. This was basically find a tissue, grind it up with a buffer, spin down the large particles, and use the supernatant. And if you look through the characteristics of those molecules or those enzyme preparations, you can see that hydrolysis efficiency was very low, purity was low, it was full of secondary enzymes and molecules and potential interferences, very high protein concentrations, and most importantly, the incubation times were very long, often greater than 3 hours, and many times we were waiting overnight to get hydrolysis done. These products do work, but have some drawbacks. Pretty much all of these enzyme preparations need some kind of secondary sample cleanup. It needs a liquid-liquid extraction, a solid phase extraction, a supported liquid extraction, a crash and spin. There has to be some cleanup to get rid of that, the gunk that is created with these preparations. Well, the Cura mission is to give you the best. Back in 2013, we introduced a product called BG100. So the development of this product took a two-part approach. First was genetic selection. Of course, there's many forms of genetic selection, but in this case, think if I need a dog to trek into the mountains and carry barrels of life-saving supplies, I look for a large, thick-coated animal with big feet to make it through the snow. Haha, St. Bernards, right? So if I want a dog to run fast, I'm looking for a sleek animal with long legs, and you immediately think of greyhounds. That was phase one. We needed to find the right animal. As it turns out, we identified a Chilean-sourced red abalone that's grown in a certain region with a certain estuary flow, harvested at a certain time of the year. The second part of this improvement was in purification. I can't give you the secrets, but Cura has a proprietary purification process that greatly improves hydrolysis efficiency. The purity is better because we remove vastly more contaminants, also impacting the levels of secondary enzymes and protein concentrations. The biggest impact realized for the laboratory is incubation times. Before, we always spoke in how many hours it took to do hydrolysis. When we started working with BG100, we always talk about the numbers of minutes that we're going to use. So here's some data from 2014 by Gregory Janis and Dr. Tan from MedTox comparing BG100 to other enzymes. And you can see a dramatic difference in the time needed to hydrolyze codeine 6-glucuronide. Codeine 6-glucuronide, we believe, is the hardest compound that we currently deal with in our panels. So here we can see that at 60 minutes, BG100 has a recovery of between 70 and 80%, while the other molecules are still below 20%. Now the INSAT shows that these other enzyme preparations do work, but you have to note that it goes to 24 hours to get some of these lines to pop up. Here's another look at how BG100 performs across a different group of molecules. So we've got a benzo, we got THC, buprenorphine, morphine, and codeine. and we look at recovery, trying to achieve greater than 95%, we can see that we get there in, it takes about 60 minutes. Of course, amitriptyline and cyclobenzaprine are not very well cleaved. So it's not a perfect system, but it does work better than what we've had before. So today, we kind of talked about the first level native enzymes and the difference between that and Kura Biotech's BG100, which gives us greatly improved hydrolysis efficiency, better purity, and significantly improved incubation times. Now, here's the teaser for tomorrow. You got to tune back in and learn how we turn all of the blocks green by using recombinant enzymes. So we look forward to seeing you tomorrow. Hit us up on your favorite social media, and cheers.